Good evening, folks. Wow, are we too loud? Are we good? All right. Okay. Well, thanks for being here. I really appreciate you coming. And I will just say a few words. I am a professor at Arizona Christian University. And if you want your children, grandchildren to go to a university where the teaching is solid, it's focused on Scripture, and in all of the various majors, whether it be communication, whether it be biology, pre-law, whatever, the focus is on the truth in Scripture. No compromise. So keep that in mind as a place for students to go if you want them to have a solid education that will be Christ-centered. And you can't say that about all Christian universities. A lot of them are very compromised. Alrighty, valuable and vicious viruses and a vaccine. So first I'll give a disclaimer that what I have to say is not necessarily the policy or viewpoint of this church or of ACU, although I think it really is, but I can't say that. All right, so we're going to talk about viruses, but I have to explain where they belong in the order of things living. So we're going to talk a bit about bacteria as well, because the two are very closely linked to each other. Viruses can infect every type of cell, but they are not living things, but they have to get inside of a cell to be reproduced. They have to take over the machinery of the cell in order for the virus to be reproduced. So the virus is not living, it's lifeless, but it's more than just a lifeless molecule. It's kind of a weird thing in between. So the best way to think of them is infectious particles. To give you an idea of the scale of the size of viruses, compare them to these two bacteria. The one on the top left is Streptococcus, the one that's known for causing strep throat. All right. The one on the top right is E. coli, the one that's known for being a common cause of infections in the urinary tract, the bladder, but also is a normal inhabitant of the gut. So the relative sizes there, and then you look at number 9, that's the size of the coronavirus we've been dealing with for the last year and a half or so. So you can see that the virus is very, very tiny, and then these other sizes are other viruses as well. So I won't bug you with these numbers, but to give you a comparison here, a sheet of normal copy paper is about 50 times wider than the virus. Okay, just to show you how you can relate it. Or the other thing is, the thickness of an average sized human hair is about 50 times wider. All right? Viruses come in 4 basic shapes, either spheres, rods, or a 20-sided figure, or this thing that kind of looks like a space lander here. And there it is in more sharper detail, a picture taken by a beam of electrons, an electron microscope, which shows things much smaller than we can see with a light microscope. So here is a colorized version of that in a diagram form, and it does look like one of these space landers, right? So let's take off and see what happens, what we have for you here. So let's first of all talk about the evolutionary ideas about the existence of viruses, how they came into being. There's 3 theories, and it's the idea, the same thing with the common cold. None of them work, all right? So that's why there's more than one. So here is a basic plan of a bacterium. And you see there's various parts, the genetic material inside that's either DNA or RNA. And then you see these various protective layers around it, and there's also a whip or flagellum so it can move through the liquid medium that it's in. And so one of the theories about viruses that the evolutionists have is that much of this was lost, and that the first living thing was a bacterium, and then the viruses devolved from bacteria. That's one of their theories, that it lost the other parts, so that the only thing left is either the RNA or DNA inside and that protective protein coat called a capsid. So those are the two basic parts of a virus. All right, another theory is that they think the viruses came first before bacteria, and that somehow this complicated molecule, RNA, came into being, and some RNA molecules actually have the ability to catalyze chemical reactions to make them happen faster. And so the word ribozyme is a combination of ribose, as in ribonucleic acid, and zyme, enzyme. So it's a combination word there. And so they like to think about all of these pre-things, pre-cell, pre-virus, pre-retrovirus, pre-ribosome. This is all wishful thinking, all wishful thinking. So they like to think that. So the virus came first before bacteria. But all of these things require information. And the evolutionists like to tell us that time plus chance equals information. But that is absolutely not true, because matter cannot give rise to information. We use matter and energy to store information, whether it be electrons in a computer, chalk on board, ink on paper, chiseling stone. So matter is used to store information, but it cannot give rise to information. So this is extremely untrue. And so the mantra, and this is a quote from an evolutionist, a very prominent one from some years ago, saying that time is the hero of the plot, and that given enough time, the impossible becomes improbable, the improbable becomes possible, and the possible finally happens. But that's not the way it works in real life. Then the third thing is, there are genes in our cells, in our nuclei of our cells, that move around inside the nucleus of the cell from one place in the DNA to another place in the DNA. And those were nicknamed jumping genes when they were first discovered, and they were assumed to move around in random chance fashion, due to ignorance and arrogance combined on the part of the evolutionists. But with further study, it became determined that these genes are moving around in extremely specific patterns to turn other genes on and off in response to changed conditions that the cell is finding itself in. So the proper name is transposon, trans meaning across, poson, position, moving across from one place to another. And so they purposely are engineered to move around in specific response to change conditions to change other genes turning on or off to respond to what's going on. That's the real deal with these transposons. So the third idea that some evolutionists have is that they think that these transposons somehow got isolated, picked up a protein coat, and became a virus. But again, none of these work. They're all wishful thinking. And then also bacteria, in addition to the one big circular chromosome they have, in blue there on the left, they also have this little tiny small circles of DNA called plasmids. And so someone thinks that maybe these plasmids may have become a virus. But again, all of these are not real. Well, what's the biblical model? In other words, why did God create viruses? Well, just to give you a picture of what we're dealing with here, it's estimated, and again, I have to talk about bacteria to talk about viruses so you understand the bigger picture. Estimated that there are anywhere from 10 million to 1 billion different kinds of bacteria. That's a huge number. Okay, we only know about a few thousand of these things, or maybe several thousand, but there's so many of them out there. And bacteria are the base of our life support system on this planet. So in the terrestrial environments on land, bacteria are the basis, that protozoa and other things that are bigger than bacteria eat them, and then bigger things in turn keep eating them, the food chain. Well, this is true in the land as well as in aquatic environments, whether it be the ocean or lakes or ponds or streams, rivers, that begin, bacteria in the black box are the basis, and they digest and eat up organic material from, let's say, a dead fish or whatever, or a plant that is rotted in the water, and they in turn are food going up the food chain. So again, bacteria are the basis of everything, but they have to deal with viruses. They're very intimately connected. So it's estimated that the number of viral species that affect multicellular organisms, in other words, organisms that are more than just one cell, would that be plants, animals, or even fungi? We have an estimation of 100 million of those. It's estimated the number of viral species that affect bacteria themselves is so high that they don't have a clue. Huge number. And next time you go swimming in that nice Pacific Ocean, just think about that there are about 200,000 species of virus in that ocean, and there are trillions of viruses in every teaspoon of that ocean that you're swimming in. Enjoy. But don't worry about it, because those viruses are not interested in you. Because the interaction between virus and a cell is extremely specific. And those viruses are not interested in our cells, so you're okay. Well, so what is the role of viruses in a biblical creation model? We said that bacteria are the basis of the life support system of the food chain, both on land and in water. Well, viruses are seen as acting in the same way a honeybee acts. Honeybees will pollinate, going from one flower to another flower, bringing the pollen from one to another so germination can happen, and then seeds can be made. Viruses are now understood to be acting in that same manner, in that they take DNA from one organism and transfer them to other organisms, whether it be from one bacteria to a different kind of bacteria or in multicellular organisms. So in the same way that bees spread pollen, viruses spread genes, which give new information to the organisms that receive those genes, allowing them to do things they couldn't do before. So that's the role of viruses. They're like trucks transporting genes with information from one organism to another one. That's their job. That's very important. So they transfer these genes, and it's now understood to be critically important. An example here is viruses can transport genes from one type of bacteria to another one in another setting, so that, for example, the gene is now acquired by the bacteria that can live in these tailing ponds and can detoxify to an extent the mercury that's present there, making it less toxic. So those bacteria that are in that environment didn't originally have that information, but the viruses can transfer those genes from one bacteria to another in a different setting. Okay? Same thing in an aquatic environment, and here it's very important because you may remember a few years ago, tuna that were caught off the Japanese coast were too toxic, full of mercury. We couldn't eat them safely. But there are bacteria that have the ability to have the genes to detoxify that mercury, and so as viruses transfer those genes from one type of bacteria to another, then those other bacteria in different situations can deal with the mercury as well. See, this is a very important part of the entire creation picture. Another example is there are certain bacteria here that eat oil spills. Alcanovorax, okay? Vorax, as in carnivore, omnivore, herbivore. Well, alcanovore, eating carbon chains. and oil. So these bacteria can eat these oil droplets and clean up these oil spills that occur. So it's not like the end of the planet when this big thing happened in the Gulf of Mexico a few years ago. These bacteria are at work breaking this stuff down. So viruses can transfer the genes that allow the bacteria to do this from one species to another species of bacteria, so that they can do that in another setting where conditions are different in the ocean, for example. So here is a quote from a secular source, University of California, Davis. If viruses were nothing but bad news, you would expect cells to be taking great pains to evolve resistance to them. And they do express resistance in some way, but they seem awfully accommodating in others. In other words, cells have specifically designed receptors to accept viruses. Very specific receptors to accept viruses. We'll get more into that. So mutations cause either no change or loss of information. There has never been a single instance ever found of a mutation causing a gain of information. Now, you've heard in the news recently this discussion about gain of function in regard to corona. That's not the same as gain of information. It's gain of function because of loss of information causing something not to happen, no longer working. So now something can happen that was inhibited before. That's how you can have a gain of function with loss of information, by taking the brake off of a particular process. Does that make sense? All right. And yet evolution is all about going from this first supposed cell that nobody can ever figure out how it happened after 100 years of research, to supposedly becoming us through so much information gained, but there is no instance of gain of information from a mutation, ever. And mutations are supposed to be the engine of evolution. But when a mutation happens, you have loss of information, and so that would be garbage in, garbage out, right? When you work with your computers, you get a bug, a virus, or something happens, it doesn't function as well, or it dies. It's the same thing. So evidence is accumulating that most, if not all, harmful viruses and bacteria are mutated forms of things that did not cause disease in the first place, non-pathogen. A pathogen is something that causes disease. Okay? So originally, just like it says in Scripture, everything was perfect and good before the fall. Viruses and bacteria were not causing disease. After the fall, mutations started happening, things started falling apart, and bad things happened. Are we there? All right. So I'll give you an example of this with the bacterium we call cholera. It's been established that it once was harmless, but became toxic after receiving this particular gene, cholera toxin gene, most likely from a virus, transferring that information. Well, the original good purpose, which still exists, is that that particular bacterium, cholera, Vibrio cholerae, breaks down the shells of dead crustaceans, lobsters, shrimp, crabs, because the ocean would fill up with these dead shells if these things weren't broken down. So the job of that bacterium is to break those things down, and the stuff can be recycled. Just like on land, if we didn't have bacteria and termites and fungi breaking down dead trees, the land would be covered with dead trees. Same idea. But what happens is, with that bacterium getting in the wrong place, as in somebody's food or drink, it causes the cells that line the gut to pump out the sodium and chloride, and with that the water follows the sodium chloride into the gut, and then people die within hours of dehydration, because all the body fluids leave and go into the gut and end up being pooped out, and the person dies unless they get rapid IV replacement therapy for the fluids. So that's a case of it being in the wrong place. So all this fits a creation, corruption, the fall, catastrophe, the flood, and confusion, the judgment at Babel, model based on biblical history. I'll tell you what, Pastor, I'm going to put into your computer this PowerPoint so folks can get access to the PowerPoint. All right? So you don't have to take your pictures, you can relax and enjoy. All right? Is that good? All right? So here's the comparison of the two models here. On the evolution side of the slide, supposedly from a very small unexplained gene pool from that first cell that somehow magically came into existence, which they cannot explain, they say that the gene pool increases due to mutations and selection. But we know that can't happen because mutations only cause no change or loss of information until change from one kind into another kind, meaning like from a cat kind into the dog kind, or cow kind to the horse kind, or ape kind to the human kind. That's what they mean by one kind into another kind, by gain of information. But we know that this is not possible. Ask any evolutionist, please give me one example of gain of information with mutation, and they cannot give you an example. Does not happen. Okay, that's a house of cards that's really actually fallen down already. It's a dead man walking. However, the creation biblical model makes a whole lot more sense and fits what we know in terms of scientific information. Because remember, the author of the Word is the author of the world. He's one and the same. He does not contradict himself. And so science, real science, can only confirm Scripture. That's the beauty of it. So from a very large created gene pool that we're told about in chapter 1 in Genesis, which divides by migration and programmed filling after the flood, after coming off the ark, then changes occur within a kind, but never from one kind to another kind. So that's how we got speciation after the flood. So that 2 cats came off the ark. Now we have 35, 36 species of cats. 2 dogs came off the ark. We have 34 species of dogs. But they're all in that dog kind. Or 8 species of bear, all in the bear kind. Or 8 species of horse in the horse kind. And it's by loss or shuffling of information, and that gets a whole talk all by itself. I can't get into those mechanisms here. And this is based on Scripture. So we have unchanging truth as the basis for this, and science is confirming it. All right, so let's talk a little bit about the anatomy of viruses. I told you that they have the DNA or RNA inside and then a protein coat, and I showed you these 4 basic shapes: the rod, the sphere, the 20-sided figure, and the space lander, right? Okay, those are actual photographs taken with electron micrographs, but they've been colorized, because always the photographs from electron micrographs are either black and white and gray. All right, so those are the 4 basic shapes. The name for the one on the far right is bacteriophage. That literally means bacterial eater. That's what it means, because that kind eat bacteria. So here's that basic structure, the DNA or RNA inside with the protein coat. And an example here showing with the rod-shaped ones is that multiple protein molecules make up that coat that protects the RNA or DNA inside. Or, for example, with a spherical one, and then you see the 20-sided one up there top left. And then you see a mention here of an envelope. Well, that's because when the viruses have been replicated, copied inside of a cell that they infected and took over the machinery, when they exit that cell, some of them take part of the membrane of the cell with it as they leave. And that's how it acquires the envelope. The envelope originally is the cell, host cell material that they acquire as they leave. Well, that envelope then is the protein part of the envelope is then made up of the viral proteins, replacing most, if not all, of the regular membrane proteins of the host cell. So you see here this mention of these various parts here. We'll go through them. So that is the envelope taken from the host cell that was infected by the original virus. It's two layers of molecules that are fat type molecule that makes up the envelope. Then the viral proteins, you've heard the term spike protein a lot. Okay, and so some of these proteins in this coat do these various things. Now, I keep saying RNA or DNA is the material inside of the virus, because those are the two major categories of viruses. The ones that are made up of RNA for their genetic material have to be converted into DNA. Their RNA has to be converted into DNA so their DNA can take over the cell that they infect. Okay, so it has to be changed back into DNA to infect the cell and take over the cell's machinery. That's why it's called reverse transcriptase. Reverse because it's going not from the usual direction of DNA to RNA, but RNA to DNA first. Trans across script writing, ACE meaning an enzyme. So that word means an enzyme that is writing across in backward direction from RNA to DNA. So part of the coat is that enzyme that takes over when the cell is infected, so that the RNA is changed, viral RNA is changed to DNA, takes over, goes into the DNA of the host cell, and then takes over the machinery. Hijacks the cell. Okay, polymerases are enzymes that copy the DNA. So here you have a virus containing the reverse transcriptase that's called a retrovirus. So that coat protects the nucleic acids inside the RNA or DNA. So there's the RNA or DNA inside. There are certain proteins in that coat that attach that viral DNA or RNA to the suitable host cell. So as I mentioned earlier, there are receptors. The cell has receptors for the virus. A specific receptor for a specific virus has to dock with a specific docking mechanism on the cell that gets infected. This is what that quote meant when they said they're awfully accommodating. Okay, so it's just think of the space shuttle going up to dock with the space station. It has to have the right docking mechanism, very specific. Hemagglutinin is the name of the protein that is part of the docking process. To agglutinate, to stick to dock with. Then, once the DNA or RNA gets into the cell, then it gets incorporated, the DNA directly incorporated into the host DNA, or the RNA through the reverse transcriptase converted into DNA, so then it can be converted and then incorporated into the host DNA. All right? Then there is some of these proteins that actually stimulate the immune system of the organism that's being infected to produce antibodies that then we can have protection against it for future infections. All right? So these are all the types of proteins in that coat. All right. And then we have the proteins that are needed to detach the daughter viruses that are made from the cell as they exit the cell. So you had to have proteins to dock with the cell, hemagglutinin, and then you have to have different proteins to separate from the cell as it leaves. And that's neuraminidase. That's for the disengagement, for taking off. So here you see a series of diagrams showing the business of the daughter virus. Virion means the daughter virus, virion, leaving the cell, taking some membrane with it, and then detaching with the neuraminidase necessary to detach. Okay. And this is an electron micrograph, photograph showing that very thing happening. Colorized, but okay. So that's what so many viruses are made. Okay, I mentioned there has to be receptors. In other words, there has to be the specific receptor for the virus to be able to dock with the cell. Well, we have many, many, many different kinds of receptors on our cells doing different jobs. But there has to be a very specific one for a virus. So the virus encounters a host cell that's susceptible because it has the right docking mechanism. It then absorbs into the cell membrane, and because of this specificity, it's a limited range of cells that it can infect. So I don't know if this makes sense, but let's just say a truck labeled with Walmart can only go to a Walmart store, and its dock. Or a truck labeled with Home Depot can only go to a Home Depot store. You get the idea. All right. So here's a drawing representing this business of the coronavirus with the receptor on the cell, and you've heard a lot about spikes. This spike is part of that docking mechanism. And so there's just another diagram showing the same thing with an adenovirus. An adenovirus is one of the common cold causes. Well, as I said, it's very specific for what a virus can infect and type of cell. So parvovirus, for example, will only deal with dogs. All right? The papaya virus only with the papaya plant down in Mexico. Or swine virus, swine flu, the pig, or the avian flu, birds, tobacco mosaic virus, and then specific viruses for bats. And I'll just go ahead and say that the business with this COVID-19 thing did not just jump from bats to people. It was engineered to do so. Okay. And even more specific than not just a particular organism, but a particular tissue in the organism. So, for example, here in our upper respiratory tract, there are specific viruses that will only affect our respiratory tract and cause swelling in the airway. You've heard or have had children or grandchildren have had croup. Okay, there's certain viruses that cause that swelling in the airway, cause it being very narrow where those arrows are, and then they have that particular sound that they make when they're trying to breathe with that swelling. Okay, so the rhinovirus, rhino means nose, so the rhinovirus one of the ones that causes the common cold as well, affects these cells, and only these cells. Or we have the viruses such as hepatitis A, B, C, D, and E that only affect the liver cells. All right, so in this case I showed you hepatitis C. All right, or we have viruses only affect the lining of the gut, causing diarrhea, such as the norovirus, causing diarrhea. And then we have brain tissue here, and then the virus that causes the disease we call Zika, the Zika virus, affecting the growth of the brain in someone who is still inside the womb. So just as a review here, you see the virus is there, it has its capsid, it has the docking proteins, the host cell has the docking mechanism. So therefore, this virus can dock with this cell, it gets inside, it takes over the machinery of the cell, its DNA and RNA and proteins are reproduced. Then those proteins and the viral DNA or RNA self-assemble, so those are the virions, the daughter viruses, and then they exit using the neuraminidase to separate from the cell and may take envelope membrane from the cell to make that envelope. So from one cell infected, anywhere from 3,000 to 100,000 virions or daughter viruses can be made. So you can see how this can rapidly become a serious infection. And this can happen anywhere from 8 to 36 hours after exposure. Okay, so how do we respond to this with our immune system? Well, these are the various types of cells in our immune system. I'm only going to mention these ones in the red box because those are the ones that are the first to encounter, to deal with a virus and a bacterial or a fungal infection. Alrighty, so I'm going to specifically just talk about this one for a bit because this is the one that's necessary to be converted into either a memory cell so that after you've dealt with the initial infection, your body keeps a memory of all of the chemistry of that infection, so the next time that same thing comes around, you are ready to roll. Your factories have already been made, the assembly lines are ready just to be flipped on. And the B cells convert also into these plasma cells, and those are the factories that make the antibodies. These are what make the antibodies. These other ones, also in red, are the ones that do the first encountering, then presenting parts of the infecting thing to the B cells, T cells, and B cells so that this whole process can be triggered. So here you see in the bottom left a microglial cell, which is in the brain. That's the immune system cell in the brain, encountering something that doesn't belong there, and you can see how it changes shape to engulf it, digest it, kill it, and then present parts to the rest of the system so that the antibodies can be made. So this is the basic shape of an antibody, and the green is the variable region. This is the part that itself doesn't vary inside itself, but it's variable from one antibody to another antibody, so that it's like, let's say, having a guard, a soldier, a guard, armed person who can only guard a particular bank building in a particular location. A different guard can only guard a particular store in a particular location. So they're very specific, but that one guard does not change from one store to another, from one bank to another. Has to deal only with that one instance. The constant region never changes, and that's part of the way in which the antibody, if it's supposed to, can be attached to a cell. So everything is extremely specific. Antigen refers to what is causing the problem, the virus or the bacteria or fungus causing the problem. Okay, so how do we make so many different kinds of antibodies? Because they're so specific. One kind of antibody can only deal with the strep organism. Another one can only deal with E. coli. Another one can deal only with the particular coronavirus. Another one can only deal with the specific poliovirus, et cetera, et cetera. So how do we do it? Well, in our DNA, so that's DNA, it has parts that become sliced and diced in the production of the messenger RNA. So there you see the messenger RNA is made, and then there is shuffling. There's alternative splicing so that different combinations are made, different proteins. That green variable part of the antibody is different when the left-hand example is made versus the right-hand example. So that's how we can have a small amount of genetic information make a humongous number of different antibodies. Are we there? How many have I lost so far? Okay. The big point is everything is extremely specific. Extremely specific. We make huge numbers of different kind of antibodies that don't change from one kind to another kind, but we make zillions of different kinds. That's what you need to take home. Okay? Also, these things make other kinds of RNA, about 30 plus different other kinds of RNA. We won't talk about that. So how many different kinds of antibodies can we make? Think of it as a giant slot machine. And of all the different combinations with each individual thing, with different combinations with the others, and it's estimated we make about 10 billion different antibodies. That should blow your mind. And yet the amount of DNA necessary to do this is really minimal, really small, because of this alternate splicing, this slicing and dicing process. So antibodies are made. I showed you the part that doesn't change, so the part where it's not forked is the part that doesn't change, the constant part, so that if it need be, it can be attached to a cell. These three types are in the blood, free, floating in the blood. These two types are in different situations. The IgA are ones that are in secretions, such as mucus or breast milk, for example. IgE are the ones that are attached to cells. Those are the ones that are responsible for allergic reactions to things, foodstuffs and stuff. Okay? Pollens. All right, so the point of this is to show you the specificities, see the different shapes of each one, whether it's a round, V-shaped, or square shape. They can only interact with that same shape. So, for example, let's just say the one on the left is only good for E. coli. The one on the middle is only good for Streptococcus. So you get the idea. Specificity. So we have this army of antibodies that we make, maybe like 10 billion different ones. And we make many copies of each one, of each kind, ready to be sprung into action when needed. All right. So let's get you one more basic thing and then we'll put this all together. Valuable virus versus bacteria. Okay, we said that bacteriophages means bacterial eaters. So here it is, rounding up in good Western style. Here's an actual electron micrograph showing the viruses, the bacteriophages, attacking the bacteria, just like those spacelanders, right? That's their shape. So this is a diagram representing the injection of that either RNA or DNA into the cell so it can take over the cell's machinery. So attachment, entry of the DNA or RNA, taking over the machinery, copying itself, making its DNA or RNA and its proteins, self-assembly inside, exiting, lysis means breaking of the cell dies, and then starts all over again with the next round of cells. That should be pretty clear. Well, how can we use this? Notice I said the term valuable. How about if we use specific bacteriophages to kill bacterial infections in our bodies? That's what's going on now in terms of research. Remember, there is extreme specificity, so that virus will only attack that bacteria. It won't attack our cells. And it won't attack the good bacteria that we have in our gut and on our skin and in our upper respiratory tract. By the way, just so you know, you also have bacteria that live on your cornea, on your eyes as well. But they're doing a good job. They're keeping out bad bacteria. They're occupying the parking spaces so that the bad bacteria can't get to you. So that's why I just said, non-toxicity to us and to the bacteria that we need. And no allergic reactions to antibiotics. Antibiotics, if we use these bacteria to kill the viruses to kill the bacteria. So that's another advantage. So here it's showing that this is research going on at Mayo Clinic, for example, in situations. So who has had a knee or hip replacement here? Anybody? 1, 2, 3, 4, 5? Any more? Okay. So if everything goes well and there's no infection, you're all happy campers. But what happens if that site gets infected where they put that artificial joint in? What do they have to do? Go back in, reopen you, wash it out, clean it out, and re-close it again. Well, how about if we just stick some of these bacteriophages in there? Once we find out, get a culture, find out which bacteria it is, give you the bacteriophages, let them kill the bacteria and leave everything closed and not mess with it. That's what they're working on. Valuable virus. Okay. How about viruses with genetic disease? So this is in downtown Phoenix on 3rd Street, just north of Van Buren, downtown in the heart of a medical complex where U of A has a medical school, ASU has nursing school, NAU has a physical therapy school, and there's a cancer center, there's a bunch of stuff down there together. This building is the Translational Genomics Institute. Their job is to take the information that we've learned by sequencing the human DNA, we know this whole sequence of the bases of the ATCGs, take that information and use it to cure people's diseases at the DNA level instead of just treating symptoms. To use viruses to inject normal DNA to be able to be used to do the job that the person's DNA can't do because of mutations. That's what the job of this institute is. This is a world-class institute. It's an amazing place. Amazing place. So, why aren't bacteria already wiped out by viruses? Because I've been talking about all these bacteriophages. Because bacteria possess what are called restriction enzymes. They're called restriction enzymes because these enzymes that the bacteria own restrict the ability of the virus to attack and infect it. It's warfare, molecular warfare out there. These are a gift from God, these enzymes. Because we use these enzymes to slice and dice DNA to take a gene that we want to be made, put it into a bacteria and have that bacteria make that product of that gene for us. So these enzymes cut DNA at sites that are called palindromes. Anybody know what a palindrome is? Besides Michael. All right, you don't count. All right. Do geese see God? The answer is yes, but read it right to left. What does it say? Do geese see God? So a palindrome is something that reads the same backwards and forwards, left to right, right to left. Okay, that's a palindrome. There's some other real funny ones too. There's a bunch of them. There's almost like a whole book of these things that people dream up. So here is the example for DNA where that's the palindrome is. It's the same in the blue going that way as it is in the green going this way. All right? So these kinds of sites in the DNA are where these restriction enzymes work. So the restriction enzymes of the bacteria attack the virus and slice and dice it so it can't mess up the bacteria. But we use these using the sticky ends that are generated to have bacteria make stuff we need them to make for us. All right? So here is a section of DNA being represented. The restriction enzyme cuts out this section of DNA. The same restriction enzyme is used to open up a bacteria, and so that our gene can be put into the bacterium. And so there it is, the donor DNA in the host DNA. In this case, the donor is the human, the host is the bacteria. So what we do then is take the gene for making insulin. So when I was in the early part of my practice as an emergency physician, in those days, in the 70s and 80s, we were giving diabetics insulin from pigs and cows, because that was the only source we could get. Well, it would work, but not really super well because the body recognized those as being foreign proteins because they weren't exactly the same as human insulin molecules. The human body would make a immune system response, develop antibodies, and then the doses had to keep being increased to huge doses to still get the effect. But now, with this, we take the gene for making the human insulin using restriction enzymes, insert the gene into these sections of DNA in the bacteria, have the bacteria make the insulin, combine the two parts of the molecule together, purify it, and give it to people. So they're getting totally human insulin. No more problems with the immune system being activated. This is fantastic stuff. So now for the last 30 years or so, maybe a few less, diabetics are getting human insulin made by bacteria using the human gene because of these restriction enzymes that the bacteria have. Why? To protect themselves against the virus. Okay? So that's how viruses are valuable, triggering all that. So here it is, the gene taken out. Now we can use viruses to put the correct gene into a person who has a disease because they have a bad gene. We can use viruses themselves to be the carrier, to be the truck, to put the virus, use the virus to put the correct gene, the normal gene, into a person, delivering that virus into the right kind of cell, and then the gene is put to work making the right kind of protein and the person's disease is cured. So this is how we can use valuable viruses this way. And that's happening. That's the job of TGen, Translational Genomics Institute, is to make this happen. All right, so the proteins made then either do the job of receiving something or going out and doing something, and the problem is solved. So these are phage, meaning the virus, using it for drug and gene delivery so that things are squared away, made correct, and even to attack cancer cells. Another way a virus can be valuable is to attack cancer cells, because the markers on cancer cells change. They're not the normal markers of a normal cell. So we can make viruses specifically deal with the markers on the cancer cells. Yeah, wow. Wow is right. Okay. All right. So I'm just going to show you a few vicious viruses. Look, if you had a bacterial infection, I could prescribe what you're asking for, an antibiotic, but it clearly is a very painful virus. What might you have experienced, a very painful virus? Shingles, right? Okay, the name of that virus is herpes zoster. It is a reactivation of the chickenpox virus. So as a kid you had chickenpox, well, that virus sits in a dormant state in your spinal column for decades. Then as you get older, some stressful thing happens, different kinds of stress can trigger it, and then that virus reactivates and multiplies and follows the nerve distribution that it's infected. And so you get that kind of a rash, which is horrendous, and the rash appears after the pain, about the third day usually, and so a patient comes in and you have to, the physician has to have a high index of suspicion to diagnose it just with the history of the pain before the rash shows up, so that antiviral therapy can be started earlier and the patient can get relief earlier as well. And so it follows the distribution of the nerve. It can be both sides or it can be just one side. It can be multiple nerves. If any of you have had it, you can attest to how miserable that is. It is extremely miserable. Another one is HIV. So here is the virus docking, going into the cell and then coming out of the cell. Well, what it does is it attacks the immune system itself, the T cells, and that's the problem because then the immune system itself is wiped out. It can't make antibodies. So the person is now defenseless. Not only against this virus, but against anything else that causes an infection. And so these people get what we call opportunistic infections. They get infected by things that would not affect anybody who does not have this disease, because they are totally wiped out. And then they get certain types of cancer. One of them is called Kaposi's sarcoma, a type of skin cancer, which if you have that, then you need to check and see if they have AIDS. The Zika virus I mentioned earlier affects the brain and affects the developing baby in utero, in the womb, and causes so much damage the brain doesn't grow to proper size and end up with a small head. And these children will never be normal. And this happens inside the womb. So this is the result of what happens with this. So that baby's head is way too small for the size of the trunk. It's a real tragedy. And then there's Ebola. When this shaped virus is seen under the electron microscope, the diagnosis is 100% clear. There's no other virus shaped like this. It's called the shepherd's crook. Originated from bats in Africa. It affects these tissues, the immune system, as well as the kidneys and the liver, and it turns everything into mush, and the people bleed everywhere, every part of their body. I didn't pull the other gorier pictures off. I didn't want to freak you out, but it's bad. So here's a team taking out a dead body in their spacesuits to protect themselves, since the virus is so highly contagious. Then West Nile virus also specifically affects the nervous system, transmitted by a mosquito. Most heavily affecting here the Southwest. When it says non-human, they're talking about birds. Billions of birds have died from this virus. Fortunately, the number of people who've had significant serious infections is small, but when it happens, it can be bad news, affecting the brain. So the symptoms are pretty clear as a fact that they are affecting the brain. So he says, we're pretty sure it's the West Nile virus. Then there's the Epstein-Barr virus. Anybody here ever have mononucleosis? Mono. Yeah, it's the same virus. Same virus. Also called the kissing disease. So we know what you've been doing. Right. Well, it also causes this thing called Burkitt's lymphoma. And what it does is it causes a break in these two chromosomes, and it's always these two chromosomes in these places. So the two parts of the chromosomes swap places, and that break causes a loss of control of the manufacturing of the lymphocytes. Loss of control. So now it's a form of cancer. And it forms these hard tumors characteristically in the jaw, very interestingly. Not always, but most often in the jaw. So those are some of the vicious viruses. But we have this promise in Revelation, and God will wipe away every tear from their eyes. There shall be no more death, nor sorrow, nor crying. There shall be no more pain, for the former things have passed away. So we have this promise to look forward to and to count on. That these things that have gone wrong from the events of Genesis 3:15 with the disobedience in the Garden of Eden causing judgment to come and then things to fall apart, mutations to happen, causing diseases, viruses to do things they didn't used to do, or bacteria to do things they didn't used to do, all that will go away. So we have that to count on. And the Lord is most gracious. Questions?
And we'll start with the little wicker basket
Okay, in the back.
In line specificity, how is it that Ebola made the jump?
Okay, I'm sorry. Would you? I have a trouble.
Oh, sorry.
Repeat that.
In view of specificity holding sway, how did Ebola make the jump from bat to human when we're talking about Wuhan virus now?
Right. That's an excellent question, and I don't have the answer to that. Okay. Maybe other people do, but I don't. All right. So, and I thought about that question coming up when I was talking about it. I said, someone's going to ask me that. So, very sharp question.
So you're saying that we've got people get shingles from chickenpox. Does that also include like chickenpox vaccination? Is that later on?
Okay, good question. So the question was, can you get shingles from the chickenpox vaccination as well? All right. Up to this time, with this coronavirus thing, viral vaccinations were made by weakening a virus and giving that weakened virus as the form of vaccination. And it was called attenuated, meaning weakened, attenuated live virus. Now, they say live, but the virus never is a living thing, but they use the term live, meaning it's an intact virus that's weakened. So with that, there would be a theoretical possibility of someone actually getting the, in this case, chickenpox disease, and then later years having that happen with the shingles. And that's one of the reasons why they tried this different set of techniques with this corona stuff, is instead of using an attenuated live virus, a weakened intact virus, instead just taking specific proteins, including those spike proteins, so that the antibodies would be made to those proteins, but there would be no chance of getting the actual infection of the coronavirus from the vaccine. Did that make sense? Okay, let me say it again in another way. Up to this time, all viral vaccines were made by using an intact virus that was weakened, so that there would be a rare possibility of someone getting the actual disease from the vaccine. Okay. With this corona set of vaccines, they did not use an intact, weakened virus. They took specific proteins, isolated them, and used those to administer to people so that our bodies would make antibodies to those specific proteins. And because an intact virus was not used, there was no chance of getting the actual infection of the coronavirus from the vaccine. Does that sound better? Okay. Say again?
They're using a protein instead of a virus.
Yes, just specific proteins.
Right.
Limited number of proteins. Yes.
Create that protein?
Aha.
Identify it and create it?
Okay, the question is creating it. Okay, I would say it's pretty safe to say now there's enough evidence that those proteins were specifically modified so that they would infect people, not just bats. But
I was talking about our vaccine.
well,
They
I'm
didn't take a bunch
I'm
of
talking about the proteins they put in the vaccine.
Where did those proteins come from?
Originally the bat, but they were modified in that lab, Wuhan.
But you're talking about the virus. I'm talking about the vaccine.
Okay, that's the whole point. The vaccine is the viral protein is the vaccine.
Did they kill the virus to get the protein, or did they RNA sequence it and then
They separated out certain proteins from the virus.
So they're viral proteins.
They're viral proteins, not the intact virus. Remember, a virus is never alive in the first place.
I understand.
Okay, so they did. That's my whole point. Is this was the first time they did not use intact viruses, just specific proteins from the virus.
So they had to break the virus down.
Oh
and
yeah, oh yeah.
Find out what its protein
Yeah, we do.
sequence was, and then create the
Yeah.
protein?
Well, just simply, yeah, you could say that, yeah.
'Cause otherwise, they spend a lot of time cutting off those little things.
Yeah, yeah. You phrased it well. Yes.
So what is your opinion of the,
I'm sorry, say again?
what is your opinion of the experimental vaccine?
What is my opinion of these experimental vaccines? I think we are now beginning to find out what unintended effects are happening, and I think we should take more time to find more out before we vaccinate everybody in the world. I have not taken the vaccine myself, and I don't intend to. I think this was rushed as all part of a political program to eliminate President Trump. I think that's become pretty clear. So I think this whole thing was a manufactured situation, literally. Go ahead, do your follow-up question.
It's regarding early treatment, and I'm a retired nurse, and I
Okay.
was appalled that early treatment, we don't wait till people get stage 4 cancer before you start treating.
Again, there was so much that was said because of political reasons. Okay, you know, remember early on President Trump said, hey, what about hydroxychloroquine? What about Zithromax? Because he spoke to physicians who were forthcoming, who were being straightforward, honest. And he was murdered in the press for doing that. Well, it turns out he was absolutely correct because he was given excellent information. Also, ivermectin is, and all of these are extremely cheap drugs that have been around for decades and decades and have been used for other purposes, mainly either malaria or rheumatoid arthritis, for example. But they're well-established drugs, and they're extremely inexpensive and very easy to get a hold of and very easy to distribute and treat. Some 30 countries have done studies using, for example, ivermectin, and in India, in one of the states of India, everybody in that state was given ivermectin, and guess what? That state is free of COVID. So to say that our health bureaucracy had been politicized is an understatement. And Dr. Fauci has not served the country well. I'll put it that way. I could say it in many other ways as well. All right. Yes.
Because we've gotten such bad information
Yes.
in the last year and a half,
Yes.
I think many people are concerned that the problems with the vaccine will not be forthcoming. That information will be corrupt just as our past information.
It's a legitimate,
What are your thoughts about
that's a
that?
legitimate concern. However, there are other voices who are speaking out, and you have to not rely on the quote mainstream media. Okay? You have to rely on other sources that will be truthful, such as the Epic Times or the Conservative Treehouse or Red State. Go to those kinds of places and you'll get the real information. There's a lot of stuff that can be done. There's a group of physicians who have been very active in trying to promote the truth about these other drugs, such as hydroxychloroquine or Zithromax or ivermectin, and to get the word out to people that there are ways of dealing with this that are very inexpensive and very accessible. And I would say Big Pharma has been part of the problem as well. So, see, there are multiple interests when it comes to power and money. serving themselves and not the people. Yeah.
You were talking about so the vaccine takes certain proteins, but not all the proteins?
No, you don't need all. You just need a few. That's all you need to make antibodies to those few proteins, and then the virus will be attacked.
So is it doing that?
Well, it does it, yeah, but it's not always successful. There's unfortunately several thousand people who, after the vaccine, have gotten the disease.
How did that happen?
Well, let's just say it's not as effective as you want it to be. Also, a major problem that concerns me is there was this giant push to make sure that younger and younger people, even down to grade school kids, are being vaccinated. I think that's a giant mistake in general, because why? Young, healthy people don't get very sick with this. At Arizona Christian University, we had a fair number of the students who are on athletic sports end up getting each other infected, but they'd end up with 2 or 3 hours of fever and about 3 days of feeling a little crummy, and then maybe tired longer, and that was it. Young and healthy. Well, now there's a big reason not to vaccinate people under 30, is because now a significant percentage of them, especially the males, are getting inflammation of the heart after vaccination. And healthy people under 30 aren't going to, the cure is worse than the disease.
They get COVID and they build up antibodies and they don't need a vaccine.
Exactly. Exactly. Remember, what we're talking about here in general, now there's the exception, people who are quite elderly, people who have comorbid disease, meaning they have something else, such as chronic lung disease, asthma, emphysema, those kinds of things. Now those people probably should be vaccinated. You know, elderly with comorbid disease. But young, healthy people not. Why? Because we're dealing with something that's basically like the flu. Okay. Now, I don't diminish at one tiny bit the tragedy that's happened with those who have died from this disease. But when you look at what happened with various flu infections every year, it's not that different. And I have to tell you, the numbers that we're told, how many excess deaths there supposedly are, it's not true. I went to the CDC website, pulled down their numbers of the deaths, looking at the years. There's a beautiful graph there from 2014 on, and there's no excessive deaths. There's an increase every year because the population's bigger every year. But when you look especially at all of a sudden in 2020, there's this huge supposed spike in deaths from this virus. Well, guess what? Those numbers are the exact same numbers of people that all of a sudden supposedly stopped dying of chronic lung disease, pneumonia, and you know.
Follow the money, right?
Right. Follow the money and the power seeking. Okay. So there was tremendous misrepresentation and miscoding. So why were these people being miscoded? Well, as you said, follow the money. So the hospitals were now, with this shutdown, deprived of their usual income with the elective procedures and even just people not coming in for ongoing cancer therapy or ongoing other chronic therapy, or people who had chest pain, people who had symptoms of stroke not coming in and getting evaluated. And so the hospitals were told, okay, we'll give you compensatory money for the numbers of people who are coded as COVID deaths. So anybody who tested positive for COVID, but no matter how they died, were coded as a COVID death. Well, you can't blame, in a way, the administrators for doing that because they needed to survive financially, but it was a misrepresentation of why these people actually died. Yes, they were positive for COVID, but they didn't die from it. Some of my former students are now in either training or out, or they're in training, you know, in their residencies, And they've talked to me and they've told me, yeah, we had this young, healthy 25-year-old come in from a motorcycle accident and died from that. He was COVID positive, so he got coded as a COVID death. See? And the hospital gets enough money to survive on. So there's a lot of non-optimal ways of treating all of this information built into the system. Yes.
You're saying that the federal government said code this as a COVID death.
Yeah,
They were instructed in
Yeah.
order to get
Well,
their additional revenue.
if you want the additional revenue, if they test positive for COVID, code it as a COVID death. Yeah. Okay, yes.
One thing I found out recently is that the WHO actually redefined the definition of pandemic a few years back. So now, a pandemic, according to the WHO, is just if a virus goes from one, goes to one nation to another, multiple nations. But before, it had, I think the meaning of a pandemic was more than just how many nations we find it in. It had to do with the number of deaths. So, I'm hearing pandemic and I'm expecting to see bodies laying around, and we don't have that.
Well, you have to remember
It was
there's,
a pandemic. It was misnamed from the beginning, but I'm saying it probably was intentional for political reasons, and so this was pre-planned ahead of time, which is why they changed the definition of pandemic to give them the license to use that word so they can spread fear across the world.
the only objection I have with what you said is you used the word probably.
Sorry. That'd be nice.
Yes.
I think I had COVID back I was in California, so I actually got it early on in 2020, in March. And so would you test, is there such a thing as testing for antibodies? Is it an accurate test?
Yes, there's two different categories of tests. One for the virus itself to see if you have the virus in your body at the time, and there are other tests for the antibodies themselves. Now, will the test tell you if you have the antibody because of vaccination versus infection? No. But if you have not been vaccinated and you test positive for the antibodies, that means you had the infection.
How long does that last?
How long, that's what
Your antibodies should last you for many, many, many years. That's
they were trying to say it don't last for 90 days, that's what told me.
yeah. Like I said, there are a lot of things that are said
Yeah.
for control purposes and to beat people down.
So,
This lady was.
I could give the gentleman the phone number, or you can call us at 600 North Beeline Highway. The Healy County Health Department has a site there, and they'll test you for antibodies and for Delta, the Delta variant. The thing is, test for antibodies is just a snapshot. That's today you have antibodies.
Yeah, but
3 days later you may not.
you should have them for years.
Really?
That's the purpose of your immune
Do
system.
not degrade. Depends on your immune system, right?
Okay, the level of antibodies won't be high, but you have the ability to make more antibodies. That's what those memory cells are all about. Right, the level can go back down, but you still have the ability to make more antibodies. Okay, there was a hand over here.
I was watching the news and now they're starting to find is it the Delta variant?
They'll find whatever they want to keep you under control.
My point is it not, I've been vaccinated, is it not possible I have that virus in my nose but no illness?
Well,
It's
I
the
suppose
illness
you.
we're worried about, not whether you have the virus.
Yeah. Well, I suppose technically, if it's different enough from the original antigens, then it's possible you could get the next variant. It's possible.
But in the people they're testing,
Yeah.
they're saying they're positive for the virus,
Yeah,
even though they've been vaccinated,
yeah,
but they're not ill. So they're counting that as illness to do what you've been saying, to have power.
yeah. Control.
My son supposedly got the virus. He said no symptoms of it at all, and all that, and I'm thinking, are they actually, well, he's kind of on the line, so we'll call him that, so we can get more funding or something like that.
Well, it is, young healthy people can be exposed and not develop enough symptoms to be clinically ill. Yeah, I mean, that's what young people do.
Yeah,
Yeah, so that's very possible.
yeah,
Yeah,
latter 30s.
yeah. That's the advantage of being young and healthy.
Yeah.
Yes.
So I was just, what you were talking about earlier about you have to have, like, some people get it and some people don't. And those people have the receptors?
Well, everybody probably has the receptor. It should, well, not necessarily everybody. I'll tell you, there are genetic issues involved. For example, type A blood seems to be more at risk. Type O blood less at risk. Navajos are much greater difficulty with this than non-Navajos. So there are genetic factors, yes.
Okay, so I had COVID in February, and I donate blood, so the blood service tested my blood and said that I have the antibodies. They did a specific test, and from what I understand, I actually talked to the chief scientist of the blood service, and so it's my understanding what my body manufactured as far as my immune system is called a nucleocapsid antibody. Does that make sense?
Yeah,
Okay.
that's the antibody to the protein coat.
Okay, the coat, okay. But if I were to get the vaccine, that messenger ribolic nucleic acid string that they put in tells my cells to make a spike protein. And then my body reacts to that spike and attacks the spike, right?
Well, a spike protein is part of that nucleocapsid.
It's the outside spike part.
It's part of that protein coat.
Okay, so I have 4 kids, so I'm on my way to visit 2 of them. And one lives up in Seattle, and his wife is a physician's assistant, her dad's a medical doctor, and all medical family, you know. And they're saying you can't come without getting the shot. And I said, well, I've already had the virus, and I have the nucleocapsid antibodies that God made. I mean, you know, I'm as protected as somebody with a shot, maybe better.
I'll use a cleaner term, bullpucky.
Yeah, so they're arguing with me. No, they're not demanding. They're kind of respecting that I didn't get it, but they said you just can't come see us this summer. I mean, I've got 2 grandchildren up there, and I'm trying to reason with them, and it just doesn't make sense to me.
I've got a niece that you can't reason with, and she's a physician as well.
Right. So, you know, I mean, I'll probably just not. You know, we'll do FaceTime this. That's what I said. I guess we'll do FaceTime this summer.
Yeah, there's no need for you to get the vaccine.
No, I don't plan on. Yeah.
Can you have like adverse reactions more so if you've had COVID and then you get the vaccine? Because some people seem to have.
That is a possibility. Yes,
Could actually get.
that's a
getting
possibility. Yes.
the vaccine.
I thought I saw another one over here.
Well, I don't want to be the one around the table. We'll do one more.
Can a young person asymptomatic, they test positive for COVID, how infectious are they?
They're not. Not
what I
when they're
thought.
done with it. When they're done with it and over with, they're fine.
No, during they test positive, but they have
You're talking
no
about-
symptoms.
You talk about positive for the virus or positive for the antibody?
Positive for the virus.
Well, when they
It's
have the
very
virus,
symptomatic.
they can be infectious. Yes, during that usually 10-day period. But after that, they won't be infectious anymore.
If you enjoyed this presentation tonight, could you put your hands together a little bit? If you have any other questions, you can approach Dr. Kezele and ask him, and we're going to turn on some lights and open up the book table.